This video is on segment 4, chapter 5, pages 172 to 178. It first talks about understanding conditioned compensatory responses, and it suggests that functional nature of conditioned response. The CRs that develop during conditioning are arguably the ones that are the best for helping the organism get ready for the US. This perspective shifts how we look at conditioning. Now that we see the result of conditioning as a set of behaviors evoked by the CS that allow the animal to optimize its interaction with the US. The nature of the unconditioned response is looked at while considering the SOP theory from the previous chapter. The presentation of the US is supposed to initiate a standard sequence of events. So the US node starts in the focally active state, then it goes to A1, and then the peripherally active state in A2, and then it goes to inactive. So the two levels of activation control different responses, and if this is really the case, then there should be a, quote, response to the US that is a sequence of two responses, and one of the responses corresponding to A1 and the other to A2. It's important to remember that the CS will come to activate the US node into A2, and the response elicited by the CS should be in the second phase, not the first, in the sequence initiated. So Paletta and Wagner noticed that a morphine injection first suppresses a rat's general activity or sedates them, but then the activity is elevated above the baseline. And if these two responses correspond to A1 and A2, then the SOP prediction is clear that CS morphine pairings should allow the CS to evoke elevated activity when tested on its own. A second experiment confirmed this, and the rats received morphine injections in different contexts. The two contexts were either in the test apparatus or the home cage, and then the control group received the saline in the experimental apparatus. And in the test, the animals were injected with the saline in the test box, and when the box was associated with morphine, it displayed hyperactivity, which was the compensatory response. So the SOP would see the compensatory response as consistent with the UR. The trick to analyze the UR is to recognize that the CR will match the second component. In many cases, A1 and A2 control the same responses. Eckelboom and Stort described another way to think about the UR. They took the physiological approach, and they noted that for a response to be a UR in the usual sense, the response must be mediated by the central nervous system, which is caused by neural activity in the brain or spinal cord. And the classic responses to typical USs used in conditioning experiments are always mediated by the nervous system. The US gives input to the brain, and then the brain reacts to that input. The definition of UR gets confused when we consider the effects of drugs, the US, and because the drugs can produce many responses that we can measure. Some effects are mediated by the central nervous system in a classic way, but not others, such as insulin, which is a hormone, but in this example displays the drug theory. So the effect of insulin is usually blood glucose, and the effect occurs because insulin promotes glucose uptake to the cells. It is a direct effect that has nothing to do with the brain, and the drop in blood glucose is a new stimulus that the nervous system reacts to, and this can be dangerous. So the compensatory response is the brain's unconditional response, UR, to a peripheral input, which is the drop of blood and glucose. This leads to the compensatory conditioning of elevated blood glucose levels in the future. The compensatory responses are not only the outcome of the conditioning trials with the US, but when responses are a result of neural activity, the CR will look like the UR. Direct peripheral effects stimulate a compensation by the nervous system, and this looks like a drug opposite effect. Eckelboom and Stort perspective also allows some drugs to show conditioned sensitization effects. Drugs have multiple effects on the body. Some are nervous system mediated and some are not. The approaches predicts that a single drug may condition responses that both mimic and compensate for the URs. For example, the injections of atropine cause both pupil dilation and a drying of the mouth, and the first effect is mediated on the brain and the second is not. Ramsey and Woods noted that this is idealistic to think that one can always identify a simple CR or UR in a drug conditioning experiment. Drugs have so many effects that a given response is bound to be a complex product of a physiological process. It may be impossible to identify crucial UR and CR, and difficult to know what the crucial stimulus is that the nervous system is actually reacting to. One must predict the direction that the CR will take. Conditioning and behavior systems, the ethological biological process, has been used to understand why the form of the CR depends on the nature of the CS. Conditioning is thought to engage whole behavior systems. Behavior systems are sets of behaviors that are organized around biological functions and goals. A few examples of them are encountering a food US activates the feeding behavior system, and this could be foraging, chasing, hoarding, food handling, etc. Sexual US activates the sexual behavior system, and this could be searching behaviors, copulation responses. Frightening USs activate the defense behavior system, and animals have a way of responding to particular USs that have developed through evolution or prior learning. And once a behavior system is engaged, stimuli in the environment provide support for particular behaviors, and a CS may determine a CR in this case. Think of a system as an exhaustive set of behaviors available to that rat that generally have the function of finding and consuming food. The system is hierarchically organized so that higher functions have a number of more specific behaviors and functions that subserve them. The highest level in the system has modes. The modes correspond to the system's biggest function or goals, such as the search for food. Within each mode are modules, and they correspond to more specific subfunctions, such as general search. Each mode, module, and behavior also has its own support stimuli. For example, hunger and the onset of nightfall are stimuli that might support and initiate the general search mode. Cues would initiate associated modules and actions organized around the function of finding food. If an animal in the general search mode were to encounter a Pavlovian cue for food, an animal might change to the focal search mode, and new modules and new behaviors organized around the function of actually procuring food would occur. When food is actually presented, the animal might switch to the handling/consuming mode, and new modules and new behaviors organized around handling/ingesting of the actual food. At any point, the behavior is jointly selected by the current mode and the environment that the stimuli is in to support it. Fanslow developed an anti-predator defense system called predatory eminence. In this system, behavior is organized around the function of avoiding being eaten by another animal. Stimulus that switches the rat between the modes is the likelihood that the rat will encounter a predator. When the rat first leaves its nest, the predatory eminence is most likely very low. The rat is not likely to encounter a predator, so in the pre-encounter mode, the rat engages in behaviors such as nest maintenance, meal pattern organization, etc. And this is organized to prevent detection by a predator. When the rat detects a predator nearby, it enters the post-encounter mode, which primarily freezes the rat. If or when the predator is actually about to attack, the animal enters a circle strike mode. Behaviors are active and designed for escape or protection. Michael Doman presented the model of sexual behavior system. It involves general search mode, focal search mode, and a consummatory mode. Different types of CSs may engage in different modes, therefore different CRs. And that CS, the modes that they invoke, can potentiate behavior elicited at the next point in the sequence. Behavior systems are usually thought to be organized in time. Cues that are remote in time for the US tend to support behaviors, and they are different from cues that are more immediate, which often support consumption. Although it is common to think that increasing the interval between the onset of the CS and the onset of the US, CS-US interval, will decrease distinctive conditioning, Atkins realized that CSs with different temporal relations to the US might support different modes in the sexual system. So in conclusion, behavior systems theory gives us a useful way to think about the nature of behavior that results from conditioning. CSs don't elicit a simple unitary reflex. Instead, they evoke a whole systems or constellations of behavior. At a theoretical level, they do this by engaging pre-organized modules and modes, which are functionally organized to help the organism cope with the US. The textbook also talked about panic disorder, which is an anxiety disorder where people experience intense, out-of-the-blue panic attacks. It also talked about the interest of agoraphobia, which is literally called The Fear of the Marketplace, or Afraid of Leaving the House, and it discussed how fear conditioning is widely thought to contribute to anxiety disorders. The physiological responses that will be evoked by CS depend on interactions between processes that function overall to maintain equilibrium.